Hi everyone, my name is Andrea Han. I'm located in Seattle, Washington. My mentor's name is David Zhang, and today I will be talking about differential gene expression analysis for PAM50 breast cancer subtypes. I decided to investigate the topic of breast cancer because my grandma is a breast cancer survivor, and the prevalence of breast cancer is growing literally as we speak, and which I will talk about in my presentation. Okay, so a little bit more on breast cancer. It is characterized as the uncontrolled division of cancerous cells in the breasts. There are more than 200,000 cases per year, and some common causes are increasing age, family history, inheritance of mutations in genes like the BRCA genes, and high female hormones like estrogen. And some common treatments include surgery, medical procedures, and medications. Okay, a little bit more about the growing prevalence. So breast cancer rates have increased by 0.6% in recent years, and by 2040, there will be a global cancer burden of 28.4 cases, which is 47% higher than the 2020 cancer burden. Breast cancer is the most diagnosed cancer globally, with a total of 2.3 million new cases. It is also the fastest growing cancer in India, as it has doubled in cases from 1982 to 2005. And then now in the US, 1 in 8 women born now will be diagnosed with breast cancer sometime in their lifetime. This all illustrates the need for more effective prevention. Okay, so some more statistics about breast cancer rates. You can see in the graph on the left, the incidence rate has grown significantly since 1975, and we can also see that it takes up a big chunk of the pie chart for incidence rate at 11.7%, and yeah. Okay, so I will be investigating the genes outside of the PAM50 list, and the PAM50 genes are 50 highly differentially expressed genes used to classify breast cancer into 5 different subtypes created by genetic researchers. And these subtypes include luminal A, luminal B, HER2, normal, and basal. And these 5 different subtypes have helped oncologists decide diagnoses, prognoses, and treatment for different breast cancer because it can basically sort the type of breast cancer cell into one of these 5s, which have different characteristics. And so in my research, we are looking for highly differentially expressed genes outside of the PAM50 list because the genes in this list have already been investigated, and I wanted to see if there were any genes outside of this list that can also help define breast cancer types. Okay, so the research I did included the tumor samples from the Carolina breast cancer study on the gene expression omnibus, and they used a total of 1,219 patients, which had 1,278 breast cancer tumor samples, and they studied a total of 417 genes, and in their data set, they sorted the genes by phase, tissue, estrogen receptor status, and PAM50 molecular subtype. And so then for my research, I sorted the genes by PAM50 molecular subtype. So on the right, you can see how by sorting it in the data set, I could group the samples by their subtype like basal and HER2. And then with this, I used the gene expression differentiation analysis tool in on the GO2R website. And then I compared the gene expression in tumor samples by their PAM50 molecular subtype. So I grouped all of the samples into each of their subtype, and then I compared 2 of them at one time. So in the table here, you can see each comparison, or this is 5 out of the 10, since there's 10 subtypes. There were 5 subtypes, which resulted in 10 comparisons total. And so I compared 2 of them at one time, and then the volcano charts, you can see like the log2 fold change, which is, so if it's positive, it shows the upregulated genes, which means those are the genes that are expressed more in this subtype, and the negative log FC, which shows the downregulated genes, so these genes are expressed less. And then I also included the number of significant PAM50 genes in this table to show if the PAM50 genes were actually differentially expressed. And since there are a large number of PAM50 genes that are differentially expressed between the different subtypes, that means this is an effective way to group breast cancer subtypes. And then here are like the rest of the comparisons. And we used a Benjamini and Hochberg p-value adjustment to kind of exclude the genes that were marked significant as by random chance. And then we decided that the gene expression was significant if the p-value was less than 0.05. And so all of these genes that we marked as significant were very differentially expressed between the two subtypes, which means either one of them expressed it a lot or the other one did not express it at all, basically. From here, we took all of the significantly expressed genes and then we excluded the PAM50 gene since I wanted to focus on the genes outside of that list. And then we took the top differentially expressed genes. We took the top 10 upregulated and then the top 10 downregulated. So we can see like which subtype was expressing it more. And then we put the genes into the string protein-protein interaction networks functional enrichment analysis to look at the connections between the genes that were highly differentially expressed. Since there were 10 comparisons, I decided to only mainly look at 5. These 5 had the most significant connections. As you can see, there's a lot of lines between each of the genes. And so each little circle represents a gene that was highly differentially expressed in one of the comparisons. And then the lines in between them represented traits or functions that they were associated with that have been published in like PubMed articles or NIH articles. And so, and then after investigating these networks, like the lines in between them, I came to several conclusions about their commonly associated functions. And okay, so for the first one, this is luminal A versus luminal B downregulated. These two are the most common types of cancer and of breast cancer. And then you can see that there's a bunch of connections in between them. And the functions that these genes were associated with were microtubule cytoskeleton regulation, mitotic spindle midzone assembly, chromosome condensation, and female meiosis 1. And then moving on to basal and luminal A upregulated. Some of these functions include sister chromatid cohesion regulation, mitotic spindle assembly, chromosome and sister chromatid segregation, and aiding in the cell cycle in general. And then for normal versus luminal B downregulated, these genes help with like part of the mitotic cell cycle and chromosome condensation and organization. And for HER2 versus normal upregulated, which means these genes are more expressed in the HER2. If it's downregulated, it means they're more highly expressed in the second subtype. And so these genes have functions in chromosome condensation, mitotic cell cycle checkpoints, sister chromatid and nuclear chromosome segregation, and nuclear division. And then lastly, for basal versus normal upregulated, some of these functions include mitotic spindle midzone assembly and checkpoint signaling, chromosome condensation, mitotic cell cycle, and microtubule. And then I wanted to mention that we can't really come to a conclusion about which subtype is like the gene performs more in, because many of these genes are either inhibitors or promoters of a function. And so a gene could like promote cell division or DNA replication while another one inhibits it. But from this research, we can only conclude which subtype expresses more of a gene, not really like how much function they have. And then furthermore, the GATA3 and CECA5 genes are two genes that I noticed are very highly differentially expressed in many comparisons. And so since they were in so many comparisons, I decided to talk about them more specifically. So the GTA3 gene is highly differentially expressed in these comparisons. But if you look closer at the comparisons I listed, they are most highly expressed in luminal A and luminal B. And so its function is to control gene expression and cellular functions. And so since it is most highly expressed in luminal A and luminal B, we can conclude that these two subtypes express these the most, while other subtypes may be lacking expression of this gene, which means they're lacking control for gene expression and cellular functions. And then for the CDCA5 gene, it is most highly differentially expressed in these genes. There is not a significant pattern for which subtype it's more differentially expressed in, but some of the functions include being a key regulator of DNA repair and chromosome segregation, promoting sister chromatid association, and ensuring accurate chromatid segregation for cell integrity. And then in conclusion, there are a significant number of genes not in the PAM50 list that are both differentially expressed and in different PAM50 subtypes that play a significant role in the development of breast cancer. So basically, what I'm concluding is that the PAM50 gene list does not capture all of the genes that could help oncologists kind of decide what kind of subtype the breast cancer is. And so significant findings like this can help define further diagnoses, prognoses, and treatment for different breast cancer subtypes. And they could certainly improve the future of medicine. Thank you.
Wonderful. Thank you so much. So my first question after this presentation is, obviously you've done a lot of analysis on your own. So curious, what direction your future research will take, what if you intend to do anything more with this project or kind of spin it off into something else?
I do think this is very interesting. Obviously, like investigating genetics, you kind of need to use like coding and like online calculators like this, like the analysis tool that I used. But I do want to get some hands-on experience like in labs and stuff because I do enjoy doing research hands-on. But I think this is also a very interesting topic. I was very like lucky to be able to have a tool to use since I don't have a lot of coding experience. This was just very accessible and I really enjoyed it. So yeah.
Wonderful. And then an additional question here coming in. So what in reference to some of the charts, what's the significance of having more connections in the significant networks that you highlighted, and how might the number of connections impact the development or persistence of breast cancer?
So in the comparisons that didn't have a lot of connections, we can't really draw a lot of connections or conclusions from those comparisons since there aren't really any like common functions or like even locations of the genes and the proteins they synthesize from those genes that were highly differentially expressed. And so if there's no connections between them, it's harder to draw kind of a conclusion about the subtype. So yeah, that's basically why.
Perfect. Thank you so much. Oh, one additional question actually coming from the presenter. So what tools or softwares did you use, and what was the method for conducting the experiments?
So basically, I used the genetic samples from the Carolina breast cancer study, and then the Geo2R website that came with the dataset had an analysis tool. And so basically, what I did was essentially group the breast cancer tumors by their PAM50 molecular subtype. And then on the analysis tool, I could select all of the breast cancer tumor samples of one subtype and then group them into, like, if it was basal, I would group it into basal. And then I would select the tumor samples of like another subtype, and then it would allow me to analyze it, and then it would give the top differentially expressed genes. So if it was very expressed in one of the subtypes and then very like not significantly expressed in another subtype, it would list it as one of the top differentially expressed genes. And then I took those top differentially expressed genes and then I analyzed like their functions and their connections outside of like the PAM50 gene list. And that was basically it.
Perfect. Thank you so much.